Today I'm introducing Melissa Davis. She is an associate professor at Weill Cornell School of Medicine, and she got her start at the University of Georgia in Athens, where she got her PhD. Interestingly, doing a lot of Drosophila work. It was in genetics, right?
Yep.
And then she moved on to Yale for postdoc to Yale, and then on to the University of Chicago, where she was studying genetics, human genetics. So she went to a totally different organism. And then you went back to the University of Georgia as a faculty member, and then after that has moved up to her current position. And her work is actually really, really interesting. I met Melissa because I'm on a diversity supplement committee, mentoring committee, and she told me about her stuff. And I was like, oh, we have to have her give a talk because it's really, really interesting work on looking at genetic ancestry and disease risk with a special emphasis on breast cancer and triple negative breast cancer. I think you spend a lot of time on, which there's a much higher rate in people from African ancestry. And because of the push for precision medicine, we clearly need to take that kind of stuff into account. And, and it's a critically underserved area of research. So that was a big reason why I wanted to give her an opportunity to give a talk. So with that, please take it away.
Great. Thank you. Thank you for the invitation. I have some friends there, of course, at the institution. So I'm really happy to be here today. And bear with me, I've been away for a week at the AACR meeting. And when I came back home, my computer decided that it was upset with me for not taking it with me. So it's been kind of working kind of slow today. But hopefully you can see my screen. And if not, someone holler at me. Let's see. Good. Okay. Perfect. And just making sure I can see you as well. So I kind of like informal talks, if there's something that I'm saying that you need me to dig into a little deeper, please feel free to, stop me along the way. But today I'm going to talk about uncovering the influence of ancestry and the tumor biology of breast cancer and disparities. Required to show you my disclosures. Sometimes I like to start out just telling you a little bit about what I'm doing right now. So as was mentioned, of course, I'm an associate professor at Weill Cornell, but I also lead a diversity and disparities initiative at our Institute of Precision Medicine. I'll talk a little bit about that, or at least the work we're doing. And I serve as a cancer ethnicity scholar at the New York Genome Center, again, another sort of initiative in terms of closing the gap of genomic data that's available for diverse populations is the P1000 project, and I'm going to talk to you a lot about that. Some emerging findings, we're still really just getting ramped up for that program. But I also just want to mention here that there's a new section of a new journal called Cancer Research Communications, and the section is on disparities and equity, and I serve as a senior editor for that. And so just a shameless plug in case you guys have some work that you want to get out and disseminate across, our field. I think that this brings me to the point that over the last several years, we've started to see this explosion of utilizing genomics and genetics in the context of disparities, but it doesn't match, for instance, the focus, if you will, of disparities in the context of social determinants or socioeconomic status. So traditionally, because of the historical surveillance of minority health, most of the research that's in this space around disparities, health disparities, and cancer disparities really exists in a space of using a lens that tries to capture, for instance, the individual level rate of employment, education, income, for instance, in minoritized populations. But what I hope to show you is that through the course of some rigorous science, what we've actually been able to uncover is that there are two faces, if you will, of disparities, biological and social determinants. Now, I want to start out by addressing sort of the elephant in the room, if you will, because sometimes I feel like I'm in the crosshairs or the crossfire, if you will, from sort of this pushback to doing race-based medicine, right? So because of using race, which is an ill-defined social construct to infer biology or biological significance, historically, the utilization of race in research, medical education, and even clinical practice has sort of been hinged upon stereotypes and biases that don't really have a firm footing in empirical data, right? But what we try to do is really hone in on the questions that we want to answer. And so you may have heard it's racism, not race, right? So this is sort of the pushback that, a person's identity isn't associated with their health outcome as much as the way they are treated in society. I think that's the heart of that, sort of movement. But when you are on this side of the trenches really trying to improve the outcome and increase the survival, if you will, of patients, we have to use all of the weapons in our arsenal. And that includes looking at biological determinants that imprint on tumors. And so if we utilize the right tools for the right questions, we can work in parallel and then converge onto what we call health justice. But part of the problem of using race right now in studies is the fact that, of course, it's ill-defined. It's ill-defined, but it's also not actually captured very consistently or rigorously in any vein of society, right? So if we look at sort of the history of the United States Census, from 1970, the initial races in this nation were just white or black. You were one or the other. It was extended to include Native Americans only in 1860. And so it sort of gives this picture like the Native Americans didn't even exist until 1860, which is problematic. But we see that type of theme sort of emerge all across the spectrum. Like, for instance, it wasn't until the 1970s that Hispanic origin was introduced. It wasn't until the 2000 census that Native Hawaiians and Pacific Islanders were disaggregated from being Asian, right? And so we sort of have to fight against this othering that we see happening when we take these very broad heterogeneous groups that identify with culture, but also genetic ancestry, and disaggregate them so that we can more appropriately even capture sort of what the status is of their health over time. So what I'm going to talk to you about today is really honing on, racial disparities. So we're going to take just a minute to define what I mean by that and how it emerged in breast cancer, and then sort of the rationale that led us to looking for biological links that are associated with ancestry and triple negative breast cancer. And then I'll give you some teasers with our genomic data that's coming out, some ancestry related gene expression networks that we've identified. And then I'll close with the dark side of breast cancer. So this is a newly funded project. It's my favorite project. So saving the best for last. Hopefully I don't talk too much and not get to it. So historically, we know that African Americans, when we look at survival data about just about any cancer site in the United States, Black patients have the lowest survival in nearly all cancer types. And again, this is certainly associated with socioeconomic status in some vein, because people who live in poverty tend to not have access to preventative or curative assets to survive a cancer diagnosis, for instance. And this is a map showing sort of the intensity, if you will, of poverty across the nation by county. I'm pointing out this region more specifically because if I were to show you a population density map of where African Americans typically live in this nation, it is certainly in this region. And so this is why we tend to see high correlation between African Americans and low SES status. But more recently, in the SEER data of the status or state of cancer disparities that was released, I believe in 2019 is when this paper was published. The reference is here in the bottom. This time, what the scientists have done, the epidemiologists have done, is actually not only just stratified for race, but also affluence or poverty within race groups. And so what's interesting is that when we look at, for instance, the Black populations of breast and prostate cancer patients, regardless of even living in affluent counties, we still see a higher overall mortality in Black patients compared to their white counterparts who live in impoverished conditions. And so this tells us that while poverty does have an impact, race or being, in a minoritized race group seems to have a stronger association with lower survival than living in affluence. And this can be because of a lot of different reasons, and we'll get into a couple of those. But specifically for breast cancer, the mortality rate is 40% higher for African American women, but it wasn't always the case. So if we look across data from the mid 1970s, what we see is that white women have always had a higher incidence rate of breast cancer. However, in recent years, this is starting to converge. Conversely, prior to the mid 1980s, it didn't matter if you were Black or white, you pretty much had the same chance of surviving a breast cancer diagnosis. In the mid 1980s to the early 90s, we began to see this stark decrease in a dramatic decrease in mortality that only started in white women. And then we have this persistent gap. Now, these stars actually represent the time when targeted hormone therapies were first introduced as trial and then became standard of care. And so this was the first sort of indication that there was something distinct in these populations whereby this level of therapy was more beneficial to one population than another. Now, when we do the work of looking at the multiple risk and mediating risk factors and mediating effectors that cause racial disparities in survival, particularly in breast cancer, and this is work led by Erica Warner, we see that tumor characteristics actually has a very significant um, influence, if you will, on the survival rate. And so tumor characteristics include, for instance, the presence of hormone receptors on the tumor. So hormone receptor status actually also dictates the line of treatment. And what we find is that 67% of the subtype called hormone receptor negative, HER2 negative, or otherwise known as triple negative, tends to be more prevalent in African Americans. And triple negative breast cancer happens to also exist at the worst end of the prognosis spectrum. It's more aggressive in its phenotype. It's usually a higher grade, higher stage at diagnosis, and it tends to also be an earlier onset disease. And that early onset is also an indication that there's a genetic heritable component as well. Now, the other interesting thing about triple negative breast cancer is that it doesn't necessarily behave the same in African Americans versus white Americans. And so what I'm showing you here are the survival curves of triple negative breast cancer patients in the New York Presbyterian Hospital System, as well as the Henry Ford Hospital System, which is where my immediate previous institution was. And so what I'm showing you here are the differences of triple negative survival from screen detected versus non-screen detected diagnoses. So just generally, screen detected means that the patient wasn't aware that there was a mass in their breast, but rather because they were a part of an ongoing mammography program, the imaging actually picked up something and therefore was screen detected. Those tend to be really small and they are early stage, easy to cure. And so we expect to see a better overall survival in screen detected cases compared to non-screen detected cases, which is typically going to be patients who found the mass themselves or had other symptoms that indicated that they needed diagnostic mammography or diagnostic imaging. But now when we stratify for African American versus white American, we see this dramatic difference in survival curves between screen and non-screen detected. Now, what's also interesting about this is that the non-screen detected category doesn't necessarily mean that the patient isn't actively being screening. There's a proportion of these that are actually what we call interval cancers, which are basically what happens when, even though the patient is going for their regimented mammographies, in between those mammographies, so the interval between those mammographies, they actually find a mass. So if there was nothing there on imaging last year, but before I can get to my mammography, I feel a lump, that of course indicates a very aggressively growing mass. But when we look at sort of the raw numbers, one of the assumptions is that African American patients aren't screening as frequently as white patients. That's not actually true. We look at health services data, Black patients are as screened or more so, depending on where we are for mammography. But even more pointedly, there's twice the number of non-screen detected cases in our white population, yet they still have better overall survival. And remember that these are patients being seen in the very same healthcare systems and so have the same standard of care. And then the last note I'll make about this slide, and we'll move on, is that even looking at the screen detected cases, there's about a 10% difference in 5-year survival between African Americans and white Americans. And so this sort of speaks to whether or not therapeutic interventions are actually the same in these populations, if they're, beneficial. And for triple negative breast cancer, we still don't have a targeted therapy. And so we have to rely on systemic chemotherapies, sort of as a first line treatment. Depending on the staging, it may even also be surgery. But what I'm showing you here are two different alleles and genes that are already known from pharmacogenomic studies a decade ago or more that are associated with cardiotoxicity to, for instance, anthracycline therapies, which again are part of the first line regimen for triple negative breast cancer. So we know that these alleles are associated with cardiotoxicity to chemo, but they're not currently used in any, formative way for determining, for instance, dosing or even the type of systemic therapy for patients. But what I'm pointing out here is that the frequency of these alleles across the general population, these are not cancer patients, it's just the general world population, almost a third of individuals who are of African descent already are known to have and harbor these risk alleles for cardiotoxicity. Accordingly, we also know that African Americans tend to also present with a higher rate of treatment-related symptoms or adverse events, including neuropathies, heart disease, and typically an African American woman who would survive a triple negative diagnosis would actually have comorbidities that lead to other health problems related to the systemic therapy and the cardiotoxicity. And so why don't we already know that, and why aren't we using this data? Well, part of it is because the current data that is in existence for, clinical tools like, risk panels is severely underpowered, because the data that exists is by and large predominantly from individuals of European background, despite the fact this is not actually proportionate to the global population. I mean, there's very little representation, if you will, of African ancestry in any of the GWAS studies that is currently available. And this translates into clinic by the fact that most of the variants, or a large proportion, we'll just say more of the time, 40% more of the time, when African Americans or other groups that are non-European are tested for specific mutations and mutation panels, what's reported out isn't a lack of mutations, but actually variants of unknown significance in these genes. And that simply means that they're unactionable because we don't know what they are. They weren't seen in a GWAS study. They haven't been defined in any type of risk assessment. And so when they get sort of uncovered, if you will, in a clinical test, there's very little that we can do about it. Now, in the case of African Americans, we also know that we don't have a lot of health information, and actually there are under represented in terms of referrals for genetic testing as well. And so we take sort of this oncologic anthropology approach to really dig into whether or not there's something unique about the African diaspora that would lend to, for instance, the susceptibility, if you will, or predisposition to a certain subtype of breast cancer. And so if we consider the worldwide, distribution and incidence and mortality for breast cancer, what we find is that on the continent of Africa, it has some of the lowest incidence but highest rates of mortality on the globe, which is similar to what I just described for African Americans. More interestingly, if we look specifically at triple negative breast cancer, the highest rates of TNBC are actually in Africa, sub-Saharan Africa. And if we can separate a population based on self-reported ancestry or a racial identity that is associated with African descent, like African Americans, for instance, and Black South Africans, it's that component of the population that also has the highest rate of triple negative breast cancer. On top of this, my research partner, Dr. Lisa Newman, who's the chief of breast surgery here, also has international partners where she's been serving as a clinical trainer, if you will, in East and West Africa, observed that patients in West Africa had higher rates of triple negative breast cancer than, say, East Africa. And so we did the due diligence of actually analyzing on the continent the rate of triple negative breast cancer in nations that were able to assess this, phenotype, because it's actually not standard, for a lot of places in Africa because of the capacity to even be able to subtype for hormone receptor status. But in areas where we could find this data, the highest rates of triple negative were in West Africa. Upon my being recruited into ICS-BCS, which is the International Center for Study of Breast Cancer Subtypes, one of the first tasks that I took was to measure genetic ancestry across the cohort, and I'll describe the cohort a little bit in the next slide. But right now we have about 4,000 patients across Africa and the US in our registry. And so we took a subset of those and did genetic ancestry estimation and found that percent West African ancestry was associated with a higher likelihood of being triple negative positive, triple negative status. Now, the other part to this is that, of course, we had this mixed population. And so I was interested to see how well it actually tracks specifically with ancestry in an admixed population, right? Because it may just be sort of confounded with the lack of West African ancestry in white patients, for instance, in the broader multi-ethnic cohort. But when we looked at African Americans alone and measured genetic ancestry, again, West African ancestry was associated with triple negative status in these patients as well. Now, this is interesting because when we really think about the African diaspora, this is a very unique population, unlike any other human, population, if we consider, for instance, the transatlantic slave trade. And so in the context of sort of the distribution of genomes across the world, this isn't a typical migration pattern where there's a founder event, for instance, that would be sort of defined by what we would consider the hybrid model, where a population moves from one region to another, and then we have hybrid populations for every generation with normal genetic drift occurring. Instead, what's happening is a continuous gene flow where the African population is being dispersed through multiple generations. And this is a very significant event for the African diaspora because it occurred over hundreds of years. Millions of Africans were displaced. And so there's a huge imprint of genetic ancestry across the diaspora. So we try to capture that through our international program. Everywhere you see circles or where we have active prospective recruitment, where we capture, for instance, biospecimen of germline and tumor samples. And so by virtue of also being in the United States, we can also capture common contemporary migration patterns as well. And so currently we are working with collaborators across the US. So now everywhere we see a red circle is where we have a particular sub-cohort that sort of defines the genomic data or comprises the genomic data I'll talk to you about here in a second. But we also have this very diverse population in the New York City region, the melting pot, right? And so we harness all of this in our prospective recruitment, and we have some substantial representation of genetic ethnicity across the city. And so using funding from the New York Genome Center and the Polyethnic 1000 project, which was started by Dr. Harold Varmus and Dr. Charles Sawyers, started out initially in its phase 1 with 16 participating sites across the New York area. And the only criteria was that the patient had to have cancer and was of non-European ancestry. And so again, this is sort of the initiative of trying to fill the gap of not having enough data in the diversity pool for people of non-European descent. I serve as co-PI for the breast cancer and prostate cancer team, and our focus is really on the genomic landscape, particularly with interest around deficiency in homologous recombination. And our target population is the African diaspora in New York City, including Afro-Latinx, Afro-Caribbean, African-American, of course, and African immigrants in the city. Our pipeline involves whole genome sequencing of both the tumor and the germline, as well as transcriptomics using RNA-seq from the tumor as well. And our particular breast cohort is mainly FFPE samples and blood germline. although we do also typically use saliva. And so we have a computational sort of standard set of multi-omic approaches looking at, for instance, structural changes, both copy number variation as well as complex structural rearrangements in those. And I'll show you a cool story around that. And then the transcriptomics area, we are looking at pathways, some of which started out as, the canonical pathways that we typically see in tumors, but not necessarily behaving or being regulated in the same manner. And all of this is linked directly into, for instance, genetic ancestry. So the reason why we're really interested in homologous recombination is because specifically in triple negative breast cancer, we are aware that, the genomic test result can be informative in terms of whether or not the tumor is displaying some form of chromosomal instability that in the case of homologous recombination deficiency could also indicate that the patient would be suitable for clinical trials that inhibit the PARP1 pathway. We also know that the genetic test result is typically what's going to be used in clinic in order to indicate this homologous recombination deficiency probability, which gives the patient access to the genomic test. So if you think about this from a clinical setting, the genetic test is what is more accessible, and what would typically be shown is a pathogenic allele of BRCA that would indicate that probability of having homologous recombination, and the genomic test would show this homologous recombination. So what this is basically tracks of data that came from this publication recently using whole genome data from the somatic tumors of triple negative breast cancer patients. And what they showed was that the homologous recombination deficiency threshold was very high, and homologous recombination deficiency was very high in people who harbored pathogenic alleles in BRCA, POLB2, even RAD51C. So these are DNA repair genes. But there's a subset of these that actually don't necessarily have a pathogenic allele, but still have a high propensity to have homologous recombination deficiency. And so we're focused on whether or not we can identify if some of these would be, for instance, African Americans who have a variant of unknown significance in one of these genes. But even more interestingly, if there's another orphan DNA repair mechanism that we haven't yet defined because we haven't been interrogating these populations. Now, of course, homologous recombination deficiency and BRCA1 are also associated with the mutational signatures or the cosmic mutational signatures. Typically signature 3 would be correlated with the BRCA mutation. Another sort of observation in previous studies, we had shown that the basal-like subtype was more frequently found in African Americans, and it is the basal-like subtype of triple negative that tends to also be have the highest rate of homologous recombination deficiency. And so all of these things together really point to the question of whether or not, if we're looking in our African populations, when we find variants of unknown significance, if we can define their significance, right? So what I'm showing you right now is where we use sequence data to identify alleles across the BRCA1 gene in this case. So these are patients from Ghana, Ethiopia. And we did find this one allele that was just really high frequency in both our African patients, and it's in a coding domain. It's a variant of unknown significance. Now, when we looked at the transcriptomics of these tumors, we actually saw this upregulation or activation of the PARP1 gene, which tends to correlate with homologous recombination deficiency. We also found that people who harbored this particular mutation in BRCA also had higher overall expression. So what's driving the activation across all the tumors in our cohort happens to be the individuals who have this mutation correlated with the expression of PARP1. And so begins our quest to determine whether or not then this mutation, along with the coincidental expression of PARP1, also indicate the homologous recombination deficiency signatures, which we have not yet completed. Sorry, it's a teaser. I'm showing you, for instance, I would say our first batch of sequences off the sequencer. We have 30 now, but I can tell you that even when I update this to the number of samples that we have here, this is just showing the first 8, but even up to 30, what we found initially is that there's a higher tumor mutation burden compared to the other breast cancer cohort in TCGA. TCGA, of course, is predominantly European American, but this is also grouping all of the subtypes together. And so I think, one of the first things we need to do is sort of stratify this for different subtypes. But it's interesting just to see that the tumor mutation burden in our African and African American tumors tends to exist around the realm of the level of TMB that we would see in types of tumors that were typically driven by mutagens. So for instance, bladder, lung, and colorectal cancer. So the most mutated genes typically, and these are somatic mutations, are, primarily not that, shocking, if you will. Most of these are very well known genes. However, we do see a couple that we're really interested in determining, for instance, the actual role of how these particular mutations in the hallmark of cancer, pathways, how they actually may be operating on the tumor landscape. Another teaser. So we have measured genetic ancestry, and one of the first things we're trying to identify is an association of certain types of signatures, either with ancestry, excuse me, or with some of those variants of unknown significance. And so one of the things we've quantified, but the data didn't format appropriately. There's supposed to be a panel here that's not showing up, it looks like. But one of the things we found was that, for instance, signature 3, which I had mentioned earlier, which is associated with BRCA variants, is definitely enriched in patients who carry BRCA mutations. Again, but this is somatic. But interestingly, we also see that this signature 8 tends to be related to another allele in a different gene called ANCL1. So ANCL1 typically was replicated in GWAS studies as a protective allele, right? So, an allele is one of 2 different genotypes typically, right? So the protective allele, which we call the protective G allele, is the allele that in GWAS studies was replicated as being a protective, genotype. However, the T allele actually is a coding change, a coding variant. And what we actually found was that in our African Americans with triple negative breast cancer, they typically have the non-protective allele or the high risk allele. And when you look at the differences in the protein structure that we modeled, adding a tryptophan can really, alter the way that a protein structure changes, and certainly the way the different domains interface. This particular gene has domains that are homologous to BRCA. Previous biochemistry studies indicate that it could potentially be a DNA repair mechanism gene, but also interestingly, when we looked at the expression of this gene by survival, we found that African Americans who have high expression of the gene have lower survival compared to European Americans. And we did sort of the internal race comparisons as well, where African Americans don't have a benefit one way or another compared to European Americans who have a survival benefit. And we think this is because African Americans are expressing the altered version or polymorphic version of the protein, while our white patients are expressing the normal version of the protein. And so what's interesting is that this particular allele is associated now with signature 8. And signature 8, well, it's emerging. I can't say it for sure. Only have 30 samples. We have a target of 200. But it looks like it's possibly associated with signature 8, which also has a signature reminiscent of homologous recombination deficiency. But again, if we weren't looking in African-enriched cohorts, and if we weren't really paying close attention to sort of the translation, if you will, of GWAS studies and how it could be, confounded by the genetic background being primarily European in the GWAS study, but in patients of African descent having a completely different protein being expressed, we wouldn't be able to even start to study these things. So this is why we say there's power in inclusion and diversity, right? Even down to the basic science of tumor biology. Now, another cool thing that that's coming out of our whole genome data is sort of building on a previous study that was led by Yale Chen at Henry Ford Health Systems, where he had already shown, for instance, that copy number variations across the genome were sort of conserved, if you will, for lack of a better word, in African Americans with both breast and prostate cancer. And they were clinically relevant. And so typically what this means is that there are structural variants in the genome that could be population private. So building on that, we're now using another platform called Java, which was developed by Marcin and Malinsky here at New York Genome Center and Weill Cornell, where not only is he looking at, for instance, copy number variations that we would typically find in comparative studies, but he's also able to identify within them these complex rearrangements in both the amplification and sometimes even the deletion events that are in the genome. And so he calls these two main ones pyrgos and rigmas. And so when we start to look across our 30 patients so far, we do find a few of them that have enrichment for, these complex rearrangements, particularly in one region of the genome called the AQ24 region. Now, this is a pretty famous region because previous studies had already identified this region as a high risk region for prostate cancer initially, but it's also been shown to have risk for other types of cancer as well. Now, the conundrum in the field at the time was that these risk alleles were like in this gene desert, like there was nothing around them. There was no coding gene anywhere near them. People were looking at non-coding regions, and it just didn't tend to resolve very well. In fact, if you look across all of the studies that were trying to identify the sort of magnitude of risk from the risk alleles in the AQ24 region, and here we're seeing a meta-analysis of over 50,000 patients. Depending on what cohort, you actually saw this wide range of risk, even some with a significant amount of protection, from these alleles. And so what I think is really happening is not just the presence or absence of a genotype, but because this happens to be a hotspot for complex rearrangements, because some of those studies also noted that there was some amplification in the region, but that amplification didn't necessarily correspond with higher expression of MYC. What I think is really happening is that some of this sort of complex rearrangement also then changes the proximity of some of these risk alleles to, for instance, the enhancer region of the oncogene MYC, which is downstream. And so we're now sort of doing the calculus of when we see the presence or absence of a risk allele, can we also, using the whole genome JABA output, determine proximity of where the risk alleles are and determine whether or not the enhancer has been hijacked, for instance, in some of this complex rearrangement. And so I'm really excited about that because I think that'll open up a new perspective of how we should even be interrogating genetic risk alleles. So I'm going to switch gears now to gene expression, and this is just a reminder of the populations that we're looking at. So again, our cohorts come from dense regions of a high prevalence of African Americans across the US, including several sites in our African panel. Specifically, our initial studies really only compared African Americans versus white Americans, and this was the first study done that actually quantified genetic ancestry and utilize it as a continuous variable to identify in a linear way whether or not differential gene expression could be determined as being associated with genetic ancestry. And so prior to this, most studies only looked at self-reported race because that was all we had in terms of tools and information. But now that we have the ability to measure genetic ancestry, we actually find genes that are associated with African and European ancestry in admixed populations. But in this case, we're actually looking at comparative patients from completely different backgrounds. And so when we looked at the genes that were associated with African ancestry, as I alluded to earlier, we found these canonical pathways, and the example I show here is TP53. But interestingly, instead of it being a loss of function, what we actually found was that P53 had activating mutations, so gain of function mutations. The pathway was activated in people of African descent. And so we're following up on this in collaboration with Jill Barganetti at Hunter College, using some Ghanaian organoids, ex vivo models. So that's really cool. But in the first paper, one of the things we were trying to put forward was sort of the feasibility and the impact of looking at African ancestry in the context of tumor biology by pointing out when we looked and found these unexpected pathway relationships or even just the polarity of a canonical pathway, some of these findings could actually indicate novel targets, but not novel in the sense that it's an unknown gene. Actually, it was just novel in the sense that some of these already have drugs that are being tested or even in treatment for certain types of cancers, but that perhaps what we're finding is a mechanism that's not typical of breast cancer, but could be utilized as a potential to determine or to establish, if you will, additional chemotherapy trials specifically for breast cancer in patients who have these activated pathways. And so again, some of what we're doing to test that, we have Ghanaian models, ex vivo models, 3D organoids. We've done CRISPR-Cas9 screening for some of the kinases that were uncovered in our pathway analysis. And we're starting to get some differential selection of combinatorial CRISPR-Cas9 in drug screens. I'm not at liberty to share the details of this study right now, but we do have several other lines coming forward. And so I hope to be able to share this in the near future. In our most recent paper, we've submitted to medRxiv. Now what we're really looking at is not only just global population ancestry, which is what's shown at the top, so East Asian, South Asian, European, American, and African. We're also looking at sort of the regional level of ancestry. And what's really beautiful, in my opinion, is that when you look at, for instance, all the African Americans in our cohort, each bar here represents one patient. You can see, for instance, what I described when I said, this population is unlike any other. The amount of diversity, even in every individual, right, is very broad. So from every region of Africa, there is a component, there's a component of African ancestry from every region in Africa, just about, in most African Americans. And then even the source of the European admixture in this group tends to be very broad. And this is in comparison to, for instance, European Americans, where, their European ancestry tends to be very solid, from one location or maybe two. And then also, if you look at, for instance, this is just an anecdotal story. So in our Ethiopian patients, all of the, not all, can't ever say all, but most of the European ancestry comes from one location, which is Tuscan Italian. And so I remember that, our computational biologists were like, oh my gosh, we messed up. How is there so much European ancestry in Ethiopians, and why is it all Italian? And so Ethiopia actually touts to be the only African nation that was not colonized by Europeans. However, they have the most European ancestry. And so what that tells us from an anthropological perspective is that while they were not colonized by any, political way, they actually had a different relationship, if you will, with the Europeans. And in fact, there was an Italian colony that shared what they call a fuzzy border with Eritrea. And Eritrea was an Italian colony. And so they had a very mixed social history. And so that's why we actually see European Italian ancestry in Ethiopians so strongly. But this also kind of sets us up to look at whether or not there are distinctions in genetic ancestry associated with some of these patterns that we see in triple negative breast cancer. And so our recent paper really hones in on just looking at the African portion of our cohort, Ghanaians, Ethiopians, and African Americans, where we found, again, ancestry specific or ancestry associated gene expression patterns from African and the European background. The genes in the middle represent those that were statistically associated with both. But when we looked at the actual expression, they were in opposing polarity. So this is a set of genes that are upregulated in African, but downregulated in European and vice versa. And what I think this represents and what we're trying to do analyses for right now is a set of genes that are under regulation of a functional ancestral informative marker, such that, for instance, they could be EQTLs that are regulating the transcription of these genes in a very distinct and polar way. When we looked at the pathways that were enriched in these genes by far, and we've seen this every time we've looked in every comparative study, people of African descent tend to have a unique immunological landscape, including not only the activation of PARP1, as we described before, but we also see some immune suppressive markers that are more highly regulated or more highly expressed in people of African descent. And so for the RNA-seq portion of this study, we did deconvolution using CyberSort to estimate the level, not just the level, but estimate the quantity of certain types of immune cells. The general tumor-associated leukocyte count was higher in all of the was higher in people of higher African ancestry. Okay, the subtypes of immune cells that we identified or detected were typically B naive cells or cells that were not activated. And so we looked at this from a dichotomy of high versus low, but we also looked at it from a continuous variable. And again, some of the highest associations with African ancestry tended to be subsets of, for instance, T cells and B naive cells. We validated this through IHC by actually quantifying immune cells in the tumors. These are representative images, but typically people of West African descent had higher infiltration of immune cells compared to our Ethiopian and European Americans. And then because these were based on IHC markers, we wanted to validate, instead of using CyberSort, which tends to be an algorithm of multiple markers, we wanted to validate that we actually saw these single markers as well in our RNA-seq. And so that's what these bottom panels represent. By separation of Ghanaian, African American, or Ethiopian, we saw significant differences, for instance, in FOXP3, CTLA4. Again, these are immune suppressive markers, so we see distinct expression patterns of these markers across our patient cohort, but then also just by genetic ancestry, we see these significant differences where people of African descent have higher expression of these immune suppressive markers. And then lastly, correlating these specific markers with the CyberSort indicates that we are seeing correlation of, for instance, PD1 on T cells versus CTLA4 on other subtypes of T cells. So just indicating that the source of the immune suppressive markers is likely coming from the immune cells that are in the tumor. In the interest of time, I'm going to skip this. I'll give you the take home. Basically, we just identified that the subtypes were distinct across African ancestry, where again, we just replicated what we'd published before, which was that the basal-like subtype triple negative was more prevalent in people of African descent. And then lastly, for the transcriptomics, we identified even genes that were associated with sub-ancestry, because remember, African Americans were so heterogeneous, we wanted to see if we could actually find genes that were associated with some of the sub-ancestry groups. And again, each one of them was associated with some component of an immune or inflammatory response. So there's a African specific mutation in a gene called DARK, and the mutation is called Duffy null. It's actually a blood group. And you see the distribution of the allele, which tracks with, for instance, the continuous gene flow that I described before, but it also correlates with the incidence of triple negative breast cancer globally. And what we found is that when you look at the actual functional consequences of DARK and Duffy null mutation is a unique immune landscape. And this is because dark on red blood cells regulates the systemic inflammation that is a consequence of infectious diseases, but also the inflammation that comes from tumors. And this mutation is fixed in people of West African descent, and actually just sub-Saharan African descent because of the Bantu expansion, by virtue of selective pressure, because it confers resistance to malarial infections, specifically from Plasmodium vivax. And so coupled with the expression on endothelial cells where DARK is actually sort of transcytosing the inflammatory markers into circulation. This is supposed to be a balanced effect where it creates a gradient to recruit immune cells into infected areas. And this is why, for instance, when we looked at TCGA data, we actually saw that the higher expression of DARK in a tumor, the higher infiltration of immune cells, particularly certain types of immune cells that were correlated with the inflammatory chemokines that DARK tends to bind to and regulate. So clinically, we saw that higher levels of DARK were associated with better overall survival. And this actually tracks with the fact that higher overall counts of TALs also are associated with overall survival in both early and late stage disease. But interestingly, if we stratify this for race, African Americans completely lose the survival benefit. And this is because we think that the Duffy null phenotype creates a higher chronic inflammation that could be altering, for instance, the function, if you will, of the immune cells. And so to investigate this, we're using now imaging mass cytometry to do single cell analysis and of the DARK environment. So comparing DARK high to DARK low tumors, we have a panel of about 27 different immune markers. We're expanding this to another 6 or 7 markers to help differentiate certain macrophage subtypes and T cell subtypes. But interestingly, when we started to do the ablations and start to get the initial images from the regions of interest, one of the first things we observed was that DARK high tumors tend to have more infiltration in the solid tumor mass, where DARK low tumors tend to have isolated immune cells in the stromal region. And again, this tracked with the RNA data. On the single cell level, what we found was that there were specific phenotypes of immune cells that were more prevalent in either the DARK high or the DARK low environment. And what we wanted to start to unravel was whether or not this indicated that a DARK low tumor might have a more suppressive phenotype. And so we started to isolate specific subsets of immune cells based on the marker expression. Here I'm showing you the isolation of some of the CD8 positive T cells. Specifically, we looked then at these T cell subtypes to determine if they had high or low expression of PD-1. And again, I sort of walked into this with the hypothesis that DARK was going to be associated with more suppressive environment, but that's not the case. We actually saw we did see something that at least supported the hypothesis, which was that, first of all, DARK high tumors had many, many more immune cells, so T cells infiltrating, compared to the DARK low tumors, but these cells that were expressing PD-1 were more prevalent in the DARK high tumors. So then when I stratified for race, what I actually saw was that the T cells that had the high PD-1 expression were all from our African American patients. So this is just showing the distribution of PD-1 in this T cell subset, for instance, in the DARK high. So just showing sort of the differential quantitation of PD-1 for this subset of cells. And then the African American patients represent those blue and red cells, pseudocolored here, as opposed to the green and purple from the white patients. Now, of course, you can see some scatter from both in each, but by and large, the majority of the PD-1 positive T cells came from African Americans. And so this indicates, for instance, that when we look now in the tumor environment, in the tumor space, that the location of these exhausted T cells in African Americans compared to white Americans indicates not only a high level of infiltration associated with the presence of DARK, but particularly for African Americans, that high level of infiltration is going to be exhausted T cells. And so we think this is sort of the combinatorial effect, again, of being Duffy null as an African American and having expression of DARK on your tumor cells, leading to high levels of immune cells, but yet not having a survival benefit because the immune cells are exhausted. So to address that, and this is the last piece of data I'm going to share, I promise, we did about 200 circulating inflammatory markers using this multiplex platform. And what we've identified, we haven't added Ancestry to this yet. We only have self-reported race currently. We're still crunching the numbers for Ancestry. But one of the markers that we found associated with race, for instance, is IL-1 beta. And IL-1 beta has been shown in lung cancer to be associated with metastatic disease, growth or invasion, angiogenesis, but also deactivation of cytotoxic T cells. And we see that more prevalently in our African and African American patients. And so this chronic level of IL-1 beta could potentially be why, in a dark Duffy null patient, and any of these other markers as well that are associated with inflammation and deactivation of the activity of T cells, and even the polarization of M1s to M2 macrophages could be because of the being, Duffy null. And so in conclusion, what we think is happening is sort of a need to reestablish what the context of an immunological response looks like in a diverse patient population. Because this Duffy null mutation, again, is fixed in West Africa, Sub-Saharan Africa. Over 80% of African Americans are going to harbor this gene, and therefore have the systemic phenotype of having chronically high levels of inflammation. And so it could impact just about any disease that has an inflammatory component, but it's currently not typically part of clinical workup. And so you may see the impact of having high inflammation, but if we knew off the bat that African Americans would just simply have a different sort of trajectory of an immune response, then I think that could help mitigate some of the disparities, right, that we see currently in clinic, not just for breast cancer, but other diseases as well. And so this brings me to the conclusion of how we're trying to reach social justice in healthcare, not only by providing equitable access to the system, right, or enhancing access to the system, but fixing the system so that their equitable access gives them tools and clinical treatments that are actually appropriate for the disease that they present with in clinic, and to just have a better understanding of what the plasticity, if you will, of the disease really is. And so with that, I'll acknowledge my teams from our lab, from the Weill Cornell Precision Medicine Institute, as well as the New York Genome Center, and our funders. And we are recruiting postdocs and graduate students. So just a shameless plug there. And I'm happy to take questions if we have time. Thanks for your attention.
Thank you for that really cool talk. Yeah, we have time for a few questions.
Okay.
Does anybody have any questions? I have one, but I'll let other people go first.
I have one too.
Hi.
Hi, Melissa. Excellent talk. Thank you so much. It's just amazing all the work that you presented. And my question is related to the high tumor mutation burden that you mentioned in the tumors from African ancestry. And how can you explain or elaborate a little bit more about it in relationship with inflammation findings that you have?
Yeah.
Also exposures, to what extent we are talking about exposures, and do you see anything there in mutational signatures that can help us get more of a broader picture of what is going on?
Wonderful question. So I'll answer the first question. Well, maybe I'll answer the second question first, which is whether or not it's related to exposure. So we are starting to track, for instance, area and neighborhood deprivation, which also then allows us to geocode, for instance, environmental exposures. We haven't been able to do this for all of our patients yet, but that's certainly something that we want to investigate, right? Because again, as you just alluded to, where you live and what you're exposed to can certainly imprint, for instance, inflammation. Inflammation is also a component of what occurs when you're under a lot of psychosocial stress. So if we're talking about systemic racism from various, input across your lifetime, has already been shown to be associated with higher levels of inflammation as well. But what I think hasn't yet been done is sort of the deconvolution of just having chronically high inflammation, even in a low stress environment, which is where our, patients from other countries who are not necessarily in a structurally racist society, whether or not they also tend to have higher levels of chronic inflammation, being Duffy null, for instance, but don't typically have the same mutation burden, right? So I think that'll help us sort of unravel that aspect of things. But then comes the chicken and egg question, right? Like, is the inflammation in the tumor caused from the DNA damage, or does the DNA damage cause the inflammation? There's certainly, you know, schools of thought in both directions. And I think one way we'll get at this is through our mouse models, where we can start to what extent we can humanize the mouse and introduce some of these mutations and see if they accumulate these, you know, patterns of mutation, or, you know, if the inflammation has to come first, you know what I mean, in order to see the mutation pattern. So yeah, all work in theory in my head, but not have has not yet been done. But yeah, that's kind of the direction I would like to go. Thank you
Great,
for that
thank you.
question. It's awesome.
It sort of answered partly my question, which was, you know, one way you can get at like the signatures or the mutation load is like in vivo studies. And I was going to ask if you had started to do any of those. I know you showed some
Yeah.
data for the expression, but I was specifically interested in the mutation aspect.
Yeah, so we do have a Duffy null mouse model, and we crossed it into a transgenic breast cancer mouse. Actually, it's a C3 tag mouse, so it gets prostate and breast cancer. You know, the guys get prostate, the girls get breast cancer. But I think that, and so we're starting to do some of that imaging mass cytometry work on the tumors in those mice. But what I would really like to do is, first of all, verify that, you know, again, that systemic inflammation is occurring in those dark knockout mice. And then look at whether or not, you know, once the tumor forms, sort of what's the trajectory of the immune response. So to that extent, that's all we've, you know, yet to do. But the issue is that, of course, a knockout mouse is completely null. And African-Americans aren't dark null. It's called Duffy null, but that's a misnomer. It's actually erythrocytic null, right? So they don't have it on their red blood cells, but they still retain dark expression, you know, in other organ systems, particularly the endothelial postcapillary venules, which is what facilitates the infiltration events that are directed by dark. So that mouse model, again, on paper, but not yet, you know, established, but we could do more, you know, bone marrow derived sibling bone marrow derived sibling ablations, for instance, and try to get at that. But yeah, I'm definitely open to try and, you know, generate a mouse model that, you know, fully characterizes sort of the distribution, if you will, of the Duffy null phenotype, you know, like sort of a spatial knockout, you know, how we could do that. But the promoter for the dark gene in mice hasn't been as well defined. So we would have to be, you know, designed.
That's pretty cool stuff. I'm actually kind of interested, like if you were to look pre-cancer, like, because there's all these data out there with somatic mutations,
Yeah.
just in normal people and everybody wandering around with them, like, you know, do some of these alleles affect the rate at which, you know, like you're looking at signatures and stuff like that, you know, does that actually impact that?
Yeah.
Well, we're probably a little over time, but I just wanted to thank you again for giving this really cool talk. And
Thank you for inviting me. I'm sorry I went over. I'm sorry I had so much technical difficulty there.
that's okay.
I was going to have to pull out the backup computer for a second.
All right. Well, thank you again, and we will be in touch.
All right. Take care, everyone.